This is an A-level biology presentation looking at DNA technology and in vivo. This is part 2 of 2. If you haven't already watched the isolation presentation, please go back and watch that before you go any further. In this particular presentation, we're going to look at the stages involved in vivo gene cloning. That is after isolation. That's covered in a separate presentation. We're going to look at the different types of promoter regions and terminator regions involved in putting different genes into sections of DNA. We're going to look at transformation, marker genes, replication, plating in terms of being able to identify which microorganisms have accepted a new gene. To get started, you have to describe the 3 different methods of isolating a gene sequence. Remember, this is covered in a separate presentation that if you haven't already seen, it would be worth going back and watching again. If you do think you know the answer, pause the presentation, go with your answer and restart when you're ready to check. Well, the 3 methods of isolation are: we can use restriction endonucleases to isolate specific regions of DNA. We can use reverse transcriptase, which will mean taking messenger RNA strands to produce cDNA strands. Or of course, if we have an amino acid sequence or polypeptide sequence, we can use a gene machine to produce the DNA. So what is gene cloning and why is it important? Well, as we've said previously, gene cloning is the process of isolating and replicating a specific gene. And why is it important? Because it allows us to mass produce polypeptides. So anywhere where we're producing large amounts of polypeptide or protein for a particular purpose, for example insulin, then this is the method we would normally use to produce that polypeptide. We have already covered the first stage of isolation, and in this presentation we're going to go through the remaining stages of insertion, transformation, identification, growth and cloning. Remember, when we're doing an insertion, the gene has to be transferred using a vector. We have previously mentioned in the other presentation the purpose of sticky ends. Remember, sticky ends give you an overlapping region whenever you are cutting up DNA fragments using restriction endonucleases. Those overlapping fragments become particularly useful when it comes time to put genes back into sections of DNA, because the overlapping sections can be attached to complementary sections of the existing DNA. And the enzyme we use to stick things together is called DNA ligase. Remember, whatever restriction endonuclease we use on the isolation of the gene is the same restriction endonuclease we would use on the DNA we want to insert, because it will always cut across the same region. And because it's always cutting across the same section of DNA, the 2 sites, the gene that you've isolated and the DNA you've opened up in order to insert the gene, will always be complementary to each other. Before we can insert our DNA or gene into a new organism's DNA, normally a bacteria, we have to add what's called a promoter site, and we have to add what's called a terminator site. And the reason for this is that normal replication through transcription and translation, you have to have promoter and terminators for the ribosomes to recognize, so they know where a particular gene starts and stops. If you don't add a promoter and terminator, you will not isolate that particular gene in the new organism, and the DNA of the new organism will randomly appear as part of an insertion mutation into the existing DNA of the organism. So what you have to do is you have to make sure whatever gene you want copied has to have a promoter and it has to have a terminator, so that the ribosomes are clear on where it starts and where the messenger RNA that gets produced should terminate. We have mentioned the term vector in the presentation before, and you may remember from GCSE when talking about malaria. But a vector is basically used to transport the DNA or the gene that we want into the host cell. When it comes to bacteria, the most common type of vector is a plasmid. However, an awful lot of technology more recently has been using viruses as a type of vector. Now we're going to talk about the process of transformation. And transformation is when DNA is transferred to a suitable host. So when we get transformation, it's the process of inserting plasmids into bacteria cells, because plasmids is the most common type of vector that we use. Most of the time we'd use a combination of temperature change and calcium ions, which result in the bacteria membrane becoming permeable, and it allows the plasmids to enter. And what we've got is a transformation efficiency, which is measured in colony forming units. And basically, the higher the transformation efficiency, the more likely it is that a particular vector will get into a particular bacteria. Well, now that we've got the plasmid with the gene that we wanted inserted into the bacteria, we now have to be able to identify, of all the bacteria we've got, which ones have actually got the vector and the gene that we've inserted. So we've three types of bacteria inside our container. We will have bacteria which did not get the plasmid and did not get the gene. We will have bacteria that did get the plasmid, but the plasmid does not contain the gene, because when we were inserting the genes, that the gene did not insert. And we also have a third scenario where we have the gene that we were inserting has basically formed its own plasmid, which is completely foreign to the bacteria. So at the end of the process, we will have some of the bacteria where a normal plasmid is present, some of the bacteria with the plasmid containing the gene we require is present, which is what we want. Some of the bacteria, the plasmid from the required gene, which is that foreign plasmid, and some of them will have no plasmid, nothing at all. But the problem is, when we look at them, they all look identical. They all look exactly the same. So we have to be able to identify, of all the bacteria, which were the ones that accepted the plasmid and that plasmid contained the gene that we wanted. In order to do this, we use marker genes. And what we've got is three types of markers. We've got antibiotic markers, which means that an antibiotic does or does not work on a particular bacteria. We've got fluorescent markers, which give the bacteria certain colors, and we have enzyme markers, which means that a bacteria would perform a certain enzyme reaction. When it comes to antibiotic markers, we use a concept called replica plating technique. What we basically do is we make identical copies of all of our bacteria samples, and we make sure that one of those is completely secure and safe and kept at the side, and the other one is used for testing using antibiotics. And what we're essentially looking for is, depending on what genes and plasmids are or are not present, remember plasmids are used to provide antibiotic resistance. It's where all the antibiotic resistance genes are found. And we use the antibiotics to target and work out which of the bacteria contain the plasmid with the particular gene that we want. Well, the first group we can easily wipe out is any of the bacteria that didn't pick up the plasmid, because if they don't have the plasmid at all, then they are missing certain antibiotic resistance genes. So if we provide that antibiotic will destroy or kill any of the bacteria that did not take up the plasmid. So if you look on the screen in front of you, you've got several bacteria. We can see what's inside them just for our own purposes. But what would happen if antibiotic A is added? Pause the presentation for a moment, come up with an answer, and after 1 minute, restart the presentation. Well, what would happen if antibiotic A is added? If antibiotic A is added, anything that didn't have the gene for antibiotic A resistance would be killed off. So we would lose L, C, D, I, N, and J. If you look in the top right of the screen, you can see that the plasmid has the gene for antibiotic resistance A in the top right of the plasmid, and L, C, D, I, N, and J are all missing that particular gene. All the other samples, or the other bacteria, do have that gene, and that gene is functional, therefore they are resistant to antibiotic A. So the second part of this involves that now that we've identified all of the organisms that contain a plasmid, we now have to identify which of the ones with the plasmid also have the gene that we've inserted. And what we do is we use the marker genes, and in this particular case we're going to be looking at an antibiotic marker gene to identify which has been affected by the inserted gene. If we look here, what would happen if we add antibiotic B? Remember, look in the top right hand corner, look at where antibiotic B is, look at where the inserted gene is, and try and work out what would go on. Pause the presentation, spend 2 minutes, come up with an answer, and then restart your presentation. Well, what we've got in antibiotic resistance is we have a kind of functioning and non-functioning gene. What we've got is either the inserted gene has inserted itself into the middle of the antibiotic resistance B gene. If that has happened, the gene for antibiotic resistance B is no longer functional, but the required polypeptide that we inserted is functional. If we've got a plasmid without the gene that we wanted inserted, it does have a resistance to antibiotic B because antibiotic B is working absolutely fine. As a consequence, when we put all of these remaining bacteria into antibiotic B, anything that maintains the plasmid with the gene that we've inserted will not survive. Antibiotic B will kill and destroy that particular pathogen. And as a consequence, the ones remaining do not contain the gene that we want. And at this point, I'm hoping you spot an obvious problem, which is why we made copies and replicates of everything at the very start of the process. If we had not taken replicas plating, if we hadn't taken multiple copies, what we would have done is said, okay, well, B, F, G, M, and E, all those bacteria contain the gene that we want, but we've just killed them all off. What we can now do is go back to our second set of plates, and we can take out plate B, F, G, M, and E, knowing that they contain the plasmid with the gene that we wanted on it because they're identical copies of the ones that we have just destroyed in our test. More recently, fluorescent markers are becoming more and more common. Basically, what the fluorescent markers allow us to do is that if a bacteria contains a plasmid without the required gene, we get a certain color fluorescing through. If it contains the required gene, then we get no fluorescence coming through. So we've added genes, and we're now using other genes as markers in order to identify. And because we're using colors, it's much easier to get things automated because machines can read colors very easily, and you can quickly start automating this entire process and start doing an awful lot of sampling by machine at a much higher rate than by hand. Look at the information you've been provided on the left-hand side and use that to answer the 3 questions on the right. Pause the presentation, give yourself 5 minutes, and when you're ready, restart the presentation. Well, if you look at the left-hand side, we have 2 genes like we had with antibiotic resistance, only this time we're going to look at colors blue and yellow, and you can see that the gene is being inserted into the blue gene rather than at the bottom with the yellow gene. What color are the bacteria that do not have a plasmid? Well, if they don't have a plasmid, they'll have no color because they've got no gene for blue and no gene for yellow. What color are the bacteria with a plasmid but that it does not have the required gene? Well, if it doesn't have the required gene, that plasmid will make a yellow pigment, but it also has a blue pigment that's functional, and blue and yellow combine to make green. If, however, we have a plasmid with the inserted gene that we want in it, the yellow pigment will work absolutely fine, that gene, but the gene for the blue will not function, and as a consequence, we will have yellow bacteria, which obviously makes it much quicker and much easier to identify which of the bacteria contain our sample, and we can even take samples from those bacteria directly. We don't have to worry about replicating plates because we're not using antibiotics. We are not killing any of the bacteria in the process. We have mentioned, of course, enzyme markers, and enzyme markers work in a very similar fashion. And basically what an enzyme marker does is that a gene will produce a certain enzyme which results in a color change of different substrate. And quite often, if the gene is not present or the plasmid is not accepted, you will find that there's no color change. So for example, if you look on the left hand side, what color is the substrate if the bacteria do not have the plasmid? Well, if there's no plasmid, then you won't have any gene for enzyme 1 or any gene for enzyme 2, so the substrate remains unchanged, and in this case it starts as yellow, so it will remain yellow. If the substrate has the bacteria has the plasmid, but not the required gene, then both gene for enzyme 1 and the gene for enzyme 2 will both work, and as a consequence we'll get to that colorless state. When we have the bacteria that has the plasmid and the required gene, well, enzyme 1 gene will work fine, but the enzyme 2 gene will no longer work, and as a consequence, it will end up pink in color because it's only producing enzyme 1, the substrate's only converted to a pink substrate. We don't have that second enzyme. What I would suggest in any questions in the exam is you make a little line diagram like the bottom left that you can see and identify clearly which genes are involved in moving from step 1 to step 2 and what genes are involved in moving from step 2 to step 3, and just helps you stop getting confused. What we've got here is a question format that you could find in the exams. We have our original plate, when we soak them in antibiotic A and when we soak them in antibiotic B. And what you have to identify is which bacteria did not take up any plasmids and which bacteria contain plasmids with the inserted genes. Pause the presentation now, come up with your answer, and when you're ready you can restart the presentation. So pause now. Well, if you have no plasmid, what should happen is it says resistance to antibiotic A is controlled in gene X, and the required gene is going to be inserted into Y. So in the first instance, we're just going to look at X, because if you have a plasmid, you have gene X and you are not affected by antibiotic A, which means A, C, E, G, H, and I all contain the plasmid because they survived an antibiotic A. They must have the gene X and therefore must have the plasmid. Of those remaining, we then put it in antibiotic B, and you can see in antibiotic B that the inserted gene goes into gene which would turn Y off. As a consequence, they are not producing the enzymes needed to break down antibiotic B, and as a consequence, antibiotic B will destroy them, which means that they will not be visible, they will have been killed, and they would not appear on that plate with antibiotic B. So we are looking for plasmids that contain the gene we required. We are looking for E, H, and I. Just to finish off, you want to go back through all the different stages. You can include the isolation from the other process, the other presentation, all the way through how we get from isolated gene all the way to the end point, which is a bacteria containing a plasmid with the isolated gene on it that is able to make the particular polypeptide that we need.